Angelina Jolie has had a lot of work done. Now, I don't mean that she's had some kind of cosmetic surgery. She might have, she maybe hasn't. I honestly don't care. But what I do know is that Miss Jolie has had some pretty major medical procedures recently. She's had a radical double mastectomy. She's had her ovaries removed and her fallopian tubes removed. Now, keep in mind, this was all done not on any solid medical diagnosis of any disease, but on risk alone. She had a genetic test that shows that she has a very, very well-characterized mutation in a gene called BRCA1 that put her lifetime risk of developing breast cancer at about 87%, and in turn, her risk of developing ovarian cancer at 50%, so not great odds. But let's also layer in there that her mother died at age 56 from her own battle against ovarian cancer, and she also battled breast cancer. And this same story was repeated 7 times in Jolie's family with women. And so taken all together, her decision maybe doesn't seem so rash, but quite rational. Now keep in mind she had a big medical team helping her out. But what I would say is think about it. Think about how much that one gene has affected her family. And the question I want to pose to you all is, what if you could know the status of all the genes in your body down to the last letter of DNA? Would you want to know? So I feel honored today to be your genetic Paul Revere to tell you that whether you're prepared for it or not, your personal genome is coming. Now, what does that mean? What exactly is a genome? Well, a genome is essentially the sum aggregate of all the genes in your body, all the DNA in your body. It's a part of your anatomy, but it's a part of our anatomy we don't typically think of because we can't easily see it. So when I say the word anatomy, you're probably thinking gross structures, right? The gross anatomy, the arms, the legs, our head, our eyes, our organs. Those large structures are made up of tissues. And those tissues, in turn, are made up of an astronomical number of cells. 37 trillion cells to make any one of you. That's more cells in your body than there are stars in the Milky Way, by a lot. And these cells have their own interesting anatomy of their own down at the microscopic level. And we're not going to focus on anything really but the nucleus in the cell because that's where we find your genome. So at the highest level of organization, our DNA is bundled up into these tight little packets called chromosomes. And for humans, we have 23 pairs. Now, why do they come in pairs? Well, you got one from your mom and you got one from your dad. Now, 22 of these chromosomes are what I would call general purpose. That is, male and females have the same ones. But the last one, the 23rd one, is what we call the sex chromosome, which tells whether or not we get girl parts or boy parts. And I can tell this is a genome from a male because there's an X and a Y chromosome. And sorry, guys, but that shrivelly, tiny looking thing, that's what makes you a boy. So we all had an inherent sense that women were more complicated creatures. Well, here's your genetic proof. So these chromosomes are dense enough that we can see them under the microscope. However, to see the DNA, we'd have to unravel one of these things. And when we do, we enter a realm where we leave the ability to see it under a microscope. Now we're at the molecular level. So why is it that the cell packages up the DNA in such little tight packets called chromosomes? Well, it does so to move it around. And if you think about, you've experienced this, right? Moving an electrical cord from your front yard to your backyard, what do you do with it? Right? You wrap it around your arm. Same thing with your cell. Your cell packages it up this way to move it around. But when we unfurl it and look at it, we see this beautiful long molecule that's shaped like a twisted ladder, an alpha helix. And if we look inside that molecule, we see that it contains a language written in 4 chemical letters that we call A, T, C, and G. And that language is arguably the oldest language on this earth, dating back some 3.5 billion years to the first life for which we have a record. So DNA is unimaginably ancient. So if it contains a language, what exactly does that language say? Well, that language specifies a set of instructions. And each discrete instruction we call a gene. And a gene is just simply a stretch of DNA that contains a recipe to make a particular molecule called a protein, a different one. And proteins have their own chemical alphabet of 20 letters that are strung together in a long string that fold back on themselves to make these beautiful three-dimensional machines that carry out the chemistry of life. So if you think about it, the DNA is essentially an information storage molecule that contains instructions for making these protein machines that carry out life. Now, going from a 4-letter language of DNA to a 20-letter language of proteins is what we call the genetic code, and we cracked it. We cracked it last century. We understand it. And the very interesting thing about it is that that genetic code is universal. All life on this planet knows how to read it. Doesn't matter whether we're talking about the lowliest bacterium to plants to fish to fungus to insects to large mammals like us. It's all the same code. And to further fry your noodle, if you think about it, of all those 37 trillion cells in your body, the ones that have a nucleus, which is most of them, have an identical copy of that genome, those set of instructions. So as I mentioned before, the genome your DNA is just too small to be able to read it by looking under a microscope. So how do we read out your DNA? Well, we use a very special machine called a DNA sequencer. And what you see here is a shot from a scene down at a modern DNA sequencing facility down in San Diego, where each one of these machines is measuring a different person's genome by taking a slight little bit of you and measuring it out. And furthermore, the machines that are viewed as the best for doing this are made by a second San Diego company. So this couldn't hardly be happening more in your backyard right now. So when these machines are done, what they spit out is some information. They spit out a big long file that you can read just like a book, right to left, A, T, C, G, G, T, T. And so your genome, when we read it out, is 3 billion DNA letters long. So if you think about it, okay, well, how much computer space does that take up? Well, uncompressed, it takes up about 0.75 gigs. And now I know what you're thinking. You're like, wow, that could easily fit on my smartphone. Yes, it can. Along with the genomes of all your immediate family and say 150 of your closest Facebook friends. And you would still have room for those essential tools of modern life, like Angry Birds and Candy Crush. So now that you say, okay, well, if my genome is sitting on my cell phone or in a computer somewhere, am I okay with that? Am I comfortable sharing that information with someone else? Just how safe and secure is that? Because this could hardly be more personal information, right? Well, what I would say is that whether for good or for bad, I would peg it as about as secure as the best banking information. You might say, well, banking information gets stolen all the time. Yes, that's true. But usually when it gets stolen, it's during the transaction period. And I would say that the transactions that you're going to have with someone else with your DNA are going to be relatively few and far between. So at least it mitigates the risk a little bit. But what about once my genome gets out there and is in the world, what happens when somebody wants to choose to discriminate against me based on my DNA? And what might that look like? Well, imagine the government or my insurance company gets a hold of it and wants to deny me benefits because I have some pre-existing genetic condition. Or a potential employer finds your genome online and as they're doing their due diligence on you, looks at it and says, this person looks like they're going to be prone to get depression and type 2 diabetes, heart disease. I'm going to move on to the next gal. So at least there is one law on the books to protect against this. So back in 2008, they signed the Genetic Information Non-Discrimination Act. So there is law on the books to try to protect against the most egregious forms of this discrimination. And you might say, well, Brian, this all sounds fantastic and amazing, but why does it matter to me? Well, I don't have time to tell you about all the different ways that this could potentially impact your health care, but let's just pick one that's kind of a hot button topic, cancer. So how can this information help us against the fight against cancer? Well, if you could imagine getting your genome sequenced today while you're healthy, you've got that as kind of a baseline. And at the point at which you get sick, God forbid you get cancer, At that point, you just sequence the entire genome of your tumor and just do the 3 billion by 3 billion comparison. That's easy to do on a computer. You might say, okay, that's great, but how does that help? Well, let me give you an example of just how powerful this can be when you know. A disease called chronic myelogenous leukemia. So as many of you may be aware, leukemia is a blood cancer. It affects the blood and it affects the bone marrow. Okay, and if we look inside the blood vessel of a healthy person, this is what the scene should look like. Lots of nice looking red blood cells and the occasional white blood cell to fight off infection. Now, what can happen is we're aware of a mutation that can happen in that white blood cell. So if we go down to the level of its DNA, a change happens, a mutation happens that changes one of those protein machines. And the protein machine that gets changed in this disease is a machine called ABL. It has its molecular off switch taken away, so now it's running rampant. And this protein machine is involved with telling your cell to replicate. So your system starts filling up with these immature white blood cells that clog up your system and kill you. Now, how do we shut off a molecular machine that has lost its off switch? We can't unplug it. No, but what we can do is fashion a molecular wrench that will fit perfectly in that machine and shut it off. And if you see that yellow sticky looking thing up toward the top, that's the wrench. It fits in this machine and pretty much this machine alone. It's a drug called Gleevec. And when you give it to people with CML, they get better. Not just a little bit. You give this to these patients, they have a 98% hematological response, meaning they clear out all of those immature white blood cells. 90% of those people go on from being on death's bed to living. Now, that was just one cancer. But more and more of these cancers are falling to this directed approach, this intelligent approach of saying, I know this change has happened in the DNA, which in turn tells me which protein machines are messed up, which tells me then which molecular wrenches, which drugs to use to treat the disease. This is the way we should be treating cancer. Now think about how we do it now with non-specific chemotherapy and radiation. I know we've all known somebody who's gone through this. It's awful. It's akin to saying, look, I know you have a gopher in your backyard. I'm going to detonate this very small nuclear weapon, and it should kill that gopher, but it might mess up your house a little bit. It might kill you. So I'm hoping in time these things take over and become the norm. And we start to view non-specific chemotherapy and radiation much the way we view bloodletting now as archaic. Now, the federal government tends to be laggards with respect to science, but in this case, they've seen what's going on with cancer and now have appropriated hundreds of millions of dollars just this year for this thing we're calling the Precision Medicine Initiative, to do this same kind of approach on other diseases than just cancer, things like cystic fibrosis and others. So I'm really excited to see what this investment's going to yield. Okay, so next I want to switch gears and talk about human variation. So if we look around, we see a lot of differences in the people from around the world. We see differences in skin color, differences in the shapes of our eyes, of our lips, of our nose, differences in the darkness and coarseness and color of our hair. And what I tell you is those things that I just described are controlled by a very, very small number of genes. Most of the variation you can't even see. And historically, we've even bucketed people into these different races or ethnic categories we talk about. But I invite you to sort of look around at your neighbors. We're not that different from one another now that we've sequenced hundreds of thousands of genomes. We are greater than or equal to 99.6% genetically identical. So at least for me, this idea of race kind of breaks down a bit. In fact, I think there's a tremendous potential for consolidation. I think we could probably consolidate it down to maybe one. The human race. And look, I'm not trying to rob anyone of their heritage. No, far from it. What I want to tell you is your DNA is such a better descriptor of your heritage than any particular ethnic or racial bucket we could toss you into. That small amount of variation tells an amazing story about how our ancestors moved across this planet to basically populate every livable environment. And for people like me who want to study healthcare and medicine, it's a gold mine. One of the greatest gifts that we could give to humanity would be to measure and catalog that variation for the sake of our species. And you might say, Brian, this sounds very inspiring. I'd love to do it, but how much does it cost, right? Well, if you asked me that question when I was a graduate student back in the late 1990s, I would have said, maybe $3 billion. But this century, we've seen something amazing happen. We've seen an astronomical drop in the price. At the beginning of this century, it was a $100 million prospect to sequence any one of your genomes. But since that time, we have seen an increasing exponential drop in the cost of doing this, such that today it costs about $1,000. Something that less than 20 years ago used to cost $3 billion now costs $1,000. So look, if this has not yet reached your affordability sweet spot, just wait a little while because it eventually will. The economic barriers for thinking about doing this will be removed. And when that time comes for you, I don't want you to be afraid of this. I want you to see this as an opportunity to participate on the ground floor in what in time will be seen as the biggest sea change in medicine that we are likely to ever see in our lifetimes, as we catalog and record that variation. And the best way for us to do that is to do it together as a species. Thank you.